workaholic who never stops, not even in his sleep.
You tend to be thinking about it at all times of the day and even the night. In fact, I used to keep a pad next to the bed on the table, and I'd often wake up in the middle of the night with an idea that for an experiment, but a few of those ideas turned out to be very productive.
The teacher who inspired many.
The discovery was in the field of multivesicular body biogenesis, but his vision and the approaches that he took transformed that discovery into what is now a very general sort of phenomena in biology.
What I like the most about Scott is how excited he becomes when he figures out an idea, and that excitement is infectious. Because you're sitting with him and suddenly you'll see Scott's got this idea and he's super excited about it, and suddenly everything feels more exciting about that idea.
Scott Emmer, director of the Weil Institute for Cell and Molecular Biology at Cornell University. The 67-year-old unpacked the secrets of a mechanism that cells use to bend membranes in a unique manner. The mechanism is central to vital biological processes, including cell division and budding viruses. His landmark discovery of the endosomal sorting complex required for transport pathway, or escort pathway, provided new perspectives to research into health problems that trouble billions of people worldwide.
We never anticipated that we would uncover genes that were so specific for this unique pathway within the cell. As a basic scientist, we were most interested in just the fundamental question of how does the system work? How does the cell build this organelle? Because we use genetics, we ask the question to the organism. And by getting the mutants, the organism feeds back information by revealing the genes and the proteins that guide these processes. It was just through our simple hypotheses and then designing genetic tools and experiments that will allow us to find these mutants.
To Scott, it has always been pure joy to dig into these biological mechanisms, and his love of science dates back to childhood.
My father was manager of a factory in New York City, and my mother was a housekeeper. I had two brothers and a sister, but they encouraged whatever we were interested in. As a small child, I always enjoyed being outside and just observing things, especially the stars and planets and so forth. And we had a set of encyclopedias in our home, and I would scan through those as to, what's going on in the world, how, biology and physics and so forth. And I sort of leaned towards biology.
But science is a very wide subject, and it took some time for the molecular biologist to emerge from the student. Marine biology was one of the possibilities Scott considered in university, but he soon found out it was not for him.
In my sophomore year, we got to go out on a research vessel in the Atlantic Ocean, and it was fairly boring. It was nothing like the Jacques Cousteau shows that they showed on TV.
He decided to switch to a career that allowed him to see how the most basic biological mechanisms work: molecular genetics.
It just opened my eyes to what bacterial genetics was capable of doing, that you could, on simply using agar dishes, find mutants that exhibited defects in individual pathways. And then by characterizing those mutants, you could identify the proteins and genes that function in those pathways, and ultimately figure out the enzymatic mechanisms.
How proteins get in and out of a cell is a problem that has captivated him all through his career. This process, known as membrane trafficking, involves specific compartments within the cell. Scott's work focused on how these compartments are built. Looking into the organelle responsible for degrading and recycling of cellular waste eventually led to his landmark discovery of the escort pathway.
We wanted to understand how the cell builds this organelle called the lysosome. How do you bring the membrane and the proteins to this organelle? And so we used yeast, a simple unicellular eukaryote, where you can apply powerful genetic tools to look for mutants that had defects in the assembly of the organelle, the lysosome. And among those mutants were mutants that ultimately fell into the escort group of genes. And by characterizing the genes and their products, we slowly were able to develop hypotheses as to how the escort system might work.
Lysosome can be understood as the recycling center of the cell. Cellular waste, or damaged membrane proteins, is sorted away from the functional ones and delivered to the lysosome for degradation. Endosome is the central sorting station, one stop along the route to the lysosome. Scott explains that at the endosome, the escort machinery selectively sorts membrane proteins and encapsulates them by bending inward the endosomal membrane. The vesicles are then delivered to the lysosome, as the endosome becomes a multi-vesicular body and fuses with the lysosome. The mechanism is unique, as vesicles bud away from the cell cytoplasm, the opposite direction they normally bud in other subcellular organelles. Regulation of signaling by cell receptors and cell divisions are among some vital processes that involve the escort pathway. Since publication in 2001, there have been widespread applications of the escort pathway in cancer and neurodegenerative disease-related research, and HIV infection, in which the virus uses the escort pathway to bud and spread to new cells. Asked what enabled his achievement, Scott thanked his mentors for their great lessons and support. One of them is Randy Schekman, winner of the 2013 Nobel Prize in Physiology or Medicine.
They each, in many ways, saw things in me that I didn't see in myself. My mentors, both at Harvard, Tom Silhavy and Jonathan Beckwith, as well as at Berkeley, Randy Schekman and Jeremy Thorner, seeing how Randy oversaw the lab and offered suggestions in a way that allowed the student and the postdocs to come up with their own ideas and then pursue them. So he gave us a good deal of freedom in how we thought about our problems and developed them, but he also offered very important insights that helped to guide me as well as others in the lab into our careers.
And the most important person, Michelle, his wife of 44 years.
When there's difficult times at the lab or experiments aren't going well, when I walk in the door in the evening and come home, my wife can usually read my face immediately and be able to determine if it was a good day or a bad day of science for me. And she is very supportive all the time and has always been the strongest person and friend in my life, and I thank her very much. She's my angel.
But was it easy to keep the family together with 2 children and a spouse so devoted to his work?
Yes, it takes patience to be a scientist's wife, and at times it can be lonely when he has to stay late or if he has to travel to conferences. Taking care of kids on my own was difficult.
How did you do it?
With patience. Well, we complement each other, and opposites seem to attract. He loves his science, and I loved my teaching, gardening, and talking with my grandkids on FaceTime and reading them stories for an hour or so. But what gives me more joy is being with Scott on the weekends, traveling together. And we love each other dearly.
Till this day, Scott and his lab are still trying to learn more about the escort complexes, or the division of labor between them. He advises future scientists to look where no one else is looking to find subjects to study.
Continue to do what excites you, and as you develop your research problem, look for problems that are not the hottest thing on the market right now, but try and define a problem that you can make your own and not be competing with others.